Hi everyone. In this video, I'd like to go over the polymerase chain reaction, or PCR. So once you have the genomic DNA extracted, you can proceed to PCR. This is the next step of the DNA barcoding process. The purpose of this step is to obtain the targeted piece of DNA, which we want to use as a DNA barcode. Here you can see all the supplies we need to conduct PCR, and you can find most of them at the PCR station in the lab. We will also need PCR reagents, which we keep in the freezer at -20. You might use different PCR settings for different species, but the general protocol is the same. We use 4 primary reagents: PCR premix, forward and reverse primers, and water. The calculations in the protocols are given for 1, 5, and 10 reactions, but you can adjust this for the number of samples you need. Also, it might be a good idea to add 1 or 2 extra reactions to cover the pipetting error. When pipetting and mixing small volumes, we usually start with the largest volume and then go down to the smallest ones. So here we first pipette PCR premix in a separate microcentrifuge tube. Then we add special DNase, RNase free distilled water, and finally the primers in any order. In this demo, you can see the first step, which is adding the PCR premix and water. In this video, Anya prepares 3 reactions for 2 samples. As you can see, PCR premix goes first in the tube. And then the water should be added. Every time we need to change the pipette tip. And every time you add a new reagent, you mix it by vortexing. Okay. Next, you can see how Anya is adding the primers. Since here we use very small amounts, the primers should be carefully added directly in the mixture. And the order of the primers doesn't matter. At the end, again, you need to vortex the tube very well. After completing step 4, it's important to put all the PCR reagents back to the freezer, as they should be kept frozen at all times. Next, we need to follow the protocol and pipette 19.2 microliters of the prepared PCR mixture in each of the special PCR tubes. They are connected in what we call a PCR tube strip. Each strip has 8 microtubes, and you can break them apart if you prepare a smaller number of samples. Finally, we add 0.8 microliters of the DNA template from the sample we stored after DNA extraction. This can be tricky, and we always should be very careful to pipette and dispose such a small amount of DNA. In this video, let's take a look how PCR cocktail can be transferred to the PCR tubes first. As you remember, we have two samples here. And in this video, Anya is demonstrating how to add the DNA template. Since we have two samples here, it's important to change the pipette tips between the samples. Finally, you can see that we need to label the PCR tubes before you put them in the PCR machine. And they are now ready for PCR. We run the PCR in the PCR machine, which is on the bench near the window in our lab. For each species, we have a separate protocol, but typically any protocol runs for about 2 hours, after which the samples will hold for several hours at the fridge temperature until you come and take the samples out. That's it. Once the PCR is done, again, you can store the PCR products in the freezer. Later, you can run a gel or do DNA purification, or you can proceed immediately to DNA purification, which is a very short step, as you will see in our next video.